Okay, so I did get up the assignment 7. Just got it up before we convened this morning. So it's similar to what I've done in previous semesters, except now it's on Canvas and multiple choice. So it basically has you getting familiar with a bacterial gene system that's under transcriptional regulation and answering questions about whether this molecule is an activator, repressor, what it's controlled by, whether transcription happens under different scenarios. So might take you a little time as you're doing it to get familiar with what's going on. It's drawn from a real example, but a little bit simplified to make it easier. And just as I have a little warning in here, the color coding, if you're familiar with color coding, we were looking at red enzymes and green enzymes. I made this a brown enzyme, so it doesn't make it easier for you to guess whether it's an activator or a repressor. So every answer is possible. You got to figure out the right way. The whole thing is self-graded, so you can have multiple attempts at it, looking to get you familiar with the concepts having to do with gene expression regulation. All right. Any questions on what we've covered so far? Take a live question or a chat question. Okay. So we got up to this example, the Lac Operon. And this is kind of a good one to get familiar with. It's one that shows up, and as I'll show you a little bit later, it actually is a gene system that is used in biotechnology. So it dates way back. It's a very nice, kind of elegant example of expression regulation, all kinds of things that tie together nicely. So bacterial operons and so on. You remember what it means to be an operon? Notes up here. There we go. So I think I'll go back to the notes I had written at the end of last time. So there we go. Definition of an operon. So operon in bacteria takes advantage of something that bacteria can do, is to do transcription once and then translation independently on the different genes that are contained on this RNA. So as an operon, it's got one promoter for multiple genes, and the promoter causes transcription to happen, so that makes the RNA molecule. And in bacteria, the RNA molecule can contain instructions for 3 or more different genes. So in the Lac Operon example, we've got this LAC-Z, LAC-Y, LAC-A genes, and they're all contained in the same unit. So the same unit that gets transcribed. And then we kind of hinted at this. So expression regulation, if you regulate gene expression, in this example, basically once transcription happens, translation follows right after it. So it doesn't need a new on switch for translation to happen. The bacterial cell is going to find itself with an RNA molecule with the start codon, stop codon, those SD translation initiation sequences. If this RNA molecule exists, it's going to get translated. So the way to prevent the enzymes from being made is really stuck in that transcription step. So it's a nice system. It's a little bit complex, and I'll try to maybe simplify it down a little bit. So the Lac Operon If we focus just on the operon itself, it's got its own promoter region. That's the yellow gold color here. The promoter is the location where RNA polymerase gets attached, right? We've done that before. And if RNA polymerase can attach, then it's going to transcribe. And that question of whether RNA polymerase attaches is really the important part of regulating the gene expression. So go. Okay, so questions like, does transcription happen? Will the RNA polymerase attach to the promoter? And we saw a couple of examples, different ways that this can happen. So if we have our promoter and RNA polymerase there, one way is if the, let's say this is a repressor molecule, if that is prevented from repressing. So an inactive repressor is one way that RNA polymerase can get onto the promoter and start transcription. Another way, on there, if you have an activator, this would be a gene where the activator is required to get RNA polymerase involved. So it depends on the gene, depends on the scenario. There are many ways for genes to be regulated. These are a couple of the important ways to think about this. So if you have a gene that's under the control of a repressor, then transcription will happen if that repressor can't repress. And sometimes actually in your diagrams from the book, this is like sealed up. So this area where it normally attaches to the promoter is not available now. Can't attach to the promoter, can't repress, therefore RNA polymerase attaches and transcription happens, or an active activator. All right, good with that? Okay. So in this scenario, what's being shown here, basically this is sort of like the symbolism that the book uses for this. So the RNA polymerase has an arrow, says it can get there. It fills this dotted RNA polymerase shape on the promoter. The arrow here is active. It says transcription happens. This is showing RNA polymerase moving down the gene and making that RNA transcript. And so this is actively transcribing the gene. The other part here, this X shows that this molecule, the red molecule, can't attach to the promoter, so that makes it, the red is code for a repressor anyway from your book. But we can also tell that it's a repressor based on what it does and right here can't do. So right now it can't attach to the promoter and therefore transcription happens. So this would be an operon that is under the control of a repressor. So let's just specify here for the Lac Operon. And the repressor in this situation, I've got two diagrams here. This one shows the Lac Operon in its on position. And at that point, the repressor is kind of plugged up with this other molecule here. So what is that circle molecule that is occupying space on the repressor molecule? Flex your fingers and type something for us. Show me that you're out there. What is the purple circle? Purple circle? Purple circle? Purple circle? All right, there's one. Right. So there's a minor detail that I kind of wish they'd just simplify this down and call it one thing, but there's like two forms of lactose. So there's lactose and allolactose. Right. So technically allolactose inhibiting, but it's really the presence of lactose that is able to attach to the repressor, cause it to change its shape, and when it changes its shape, it can't interact with the promoter. So this repressor, sort of two parts to this. Figure out my clicking here. Okay, so the repressor has two shapes, and it switches between them. And this is an important, really like a key part of the regulation, is that this repressor molecule itself can be turned on or off. So if the repressor molecule is on or off, that affects whether transcription happens. And the repressor turns on or off depending on the presence of lactose. So presence, or we could say high amounts of lactose, allolactose, and in this other form, that would be low amounts. So if there's a lot of lactose around, that gets in here and interacts with the repressor, kind of plugs up this one active site, and that causes a shape change in the repressor molecule that closes up the site, the part of that molecule that would have interacted with the DNA. And if you remember this back from enzymes or maybe other topics you've covered this in, this is a form of allosteric regulation. I don't really enforce this concept in this class, but if this looks familiar to you, this is what's going on. So it's really just a molecule that has a shape and interacting with one, the lactose in this case, the signal molecule, interacting with that changes the shape of the repressor and affects its ability to interact with the DNA. So the repressor is really the thing that responds to the low amounts, high amounts, or presence or absence of lactose. So kind of a chain reaction, right? High amounts of lactose affect the repressor, prevent it from repressing, and that turns transcription on. So in the main reaction, we're finding ourselves, if you're a bacterial cell, finding yourself in the presence of lactose and breaking it down into glucose and galactose, and the enzyme responsible was beta-galactosidase, and that was encoded by the LAC-Z gene. So for this all to happen, to break down lactose, we need this LAC-Z gene to be transcribed or expressed. In this situation, the lactose, the presence of this food, is the start of a cascade of signals that allow the cell to break down that food. So it's a nice molecular, kind of a self-contained pathway. Find yourself with food, build the enzymes to break down the food, boom, energy, profit. Right? Okay. And it is kind of it's going in a circle here, right? We have the enzymes making things happen. When does it all stop? Seems like it just keeps going, right? So the arrows go, we transcribe the genes, we break down the lactose. When does it turn off? What do you think? How do you turn off transcription here? We have it in the on position. How do you turn it off? Cool. Perfect. Thank you, Amanda. Yes, the amount of lactose drops. So on the notes here, the switching between the two forms of the repressor, the lactose that's preventing this repressor from repressing, if that lactose goes away, then there's not enough to make this happen. And instead, the repressor would be in this form over here, and it'd be able to repress, able to stop transcription. And that is, it's a form of feedback, right? So let me see if I can connect the dots for us here. So we have a high amount of lactose causes transcription to turn on, causes us to digest lactose, causes the amount of lactose to decrease because it's being digested. And when this amount of lactose basically goes away, that takes away the signal that allowed transcription to happen and effectively turns it off. So bacterial cells don't have to think about this. They don't have to have a very complicated sensing system for lactose in the environment. It's all pretty self-contained there. Okay, so in the on position, I think we covered that pretty well. Over here in the off position, so this is, as I described before, this is a situation where there's no signal molecule present, and sometimes these are described as usually off. So under normal circumstances, right, lactose is kind of rare as a food source. Under normal, without lactose situation, the repressor is made, and the repressor just goes over and clamps down on the promoter. and prevents transcription from happening. So the operon is here, is kind of waiting to be called up if it needs to be, but it's mostly in the waiting inactive position. So it's clamped down by the repressor. That would make this inducible in the language we used before, right? So usually off, can be turned on, inducible. Inducible through the action of a repressor, turned on, transcription turned on in the presence of the substrate, the lactose molecule. I don't know how much detail I want to go into this. I think I have these spelled out, but I probably don't need to. I mentioned before that the repressor is an enzyme itself, and so there's a gene for the enzyme. So there's a gene called the Lac gene that makes this repressor enzyme, and it has its own promoter called P sub I. Lac P is the promoter region for the Lac Operon. I don't find it terribly helpful to consider this operator as a different element, but it's like the specific part of the promoter where the repressor attaches. So the operator is really like part of the promoter. I think that's an easier way to think about it. Okay, I think we covered those pretty well. Let's move on ahead to move on to some mutants. So in all kinds of areas of genetics, as we've seen so far, studying the functions of genes, a lot of that depends on sort of taking them away and seeing what happens, right? So when people were first learning about the Lac Operon, they would figure out these different elements and find mutants that would have a different response to the presence of lactose, different kind of enzyme performance, and try to get in there and find out, okay, what is it that has been mutated to change this? So quite a variety of mutants that we can see what they do, and understanding what the mutants do allows us to really figure out what the enzymes do that those mutants would have normally encoded for. So we'll walk through a few of these. The first one is a mutation in this Lac gene. So the Lac, as I just showed you previously, is the gene that makes the repressor molecule. So similar to what we've done in fruit flies, they're using this plus and minus notation. So plus is wild type functional version. And then this Lac minus means that this is mutated in such a way that the repressor can't do its job. So the mutation is here. That causes a repressor to be made that can't attach to the promoter. So what happens? I'll pull this up in a form I can draw on. So we've seen what happens normally. We saw that in the presence of lots of lactose, transcription is on, and low amounts of lactose, transcription is off. So what happens if we have a mutant repressor that cannot repress? Always on. Well, there you go. Right. So we saw before in high amounts of lactose, transcription would be on. The situation there was that in the normal situation, lactose would attach to the repressor and prevent it from repressing. Well, it can't repress ever. So it's mutated, can't do that part of its job. Right, so transcription always on. Great. So that tells us a lot about what the repressor is doing. If we mutate the repressor, then transcription stays on. So that must have been its job, was to turn off transcription, right? Another mutation of the Lac-I gene, this one's called super repressor. So it's called Lac-I superscript S. So if you're concerned, I'm not going to have you memorize any of the gene names or the allele names. I'll tell you what kind of mutation it is if you get this on an exam question. This one, I kind of like the way they draw it. It does have the active site that interacts with DNA. So it's able to repress, but it is unable to interact with lactose. So presence or absence of lactose doesn't matter. It's not receptive to interacting with lactose, so that means it's always repressing. So in this case, we have a different one. This so-called super repressor is always going to repress. Always going to get in the way, sit down on the promoter, and prevent transcription. Here's another one. This is a mutation in the operator, that region of the promoter. This mutation in the operator changes the DNA that normally gets recognized by the repressor and prevents the repressor from attaching. So here, in the absence of lactose, the repressor would normally attach to DNA. It's got that site at the top that normally attaches to the DNA. But because of this mutation in the Lac-O operator, it can't stop transcription. And so in the absence of lactose, transcription is on. In the presence of lactose, doesn't really matter. That would prevent the repressor from repressing, but it wasn't going to do that anyway. So transcription on all the time in this case. And just a little tidbit, we did cover this vocabulary word before, constitutive. So the Lac-O superscript C allele or version of that gene, or the not really a gene, the operator region, that turns it into constitutive gene expression. So basically always on, as we've seen in some other genes, although always on for these genes, probably not so helpful. Other types of mutations, so figuring out what all these different elements do, mutations in the LAC-Z gene, you could have transcription happen and the enzyme is made, but if you have a mutated LAC-Z gene, it's possible that the mutation causes that beta-galactosidase not to work. And that's what's shown here. So LAC-Z minus makes the galactosidase enzyme, but it can't break down lactose. So in this one, the feedback would be a little bit broken. There might be a little bit of lactose in the cell, and it would always have the signal to transcribe because that lactose is present, but it wouldn't have the ability to get rid of the lactose. And so that lactose would just stick around and keep making enzymes that can't break it down. I guess a little bit of an aside here. If you do any molecular biology, this is a technology that, or a technique that features sometimes if you are trying to add genetic elements to bacteria. They actually use the LAC-Z gene as a reporter. So you can, in this example, there's the ability to like insert genetic information sort of in the middle of the LAC-Z gene. And so if you don't insert anything, you've got a functional LAC-Z gene, makes a functional beta-galactosidase. And you can tell whether you've got functioning beta-galactosidase. It takes advantage of this artificial chemical called XGAL. And functioning beta-galactosidase turns XGAL into a blue product. So if you have the LAC-Z gene, your colonies would show up as blue. If they don't have a functioning LAC-Z gene, they come out as white. So in the examples we worked through, basically these examples, you'd be able to maybe test if transcription was happening and if this beta-galactosidase molecule was functional using this white and blue signaling system. All right, still with me so far? Doing all right? So they got pretty creative as they were investigating the Lac Operon. This investigation was looking at the effects of, so you had different versions of your repressor molecule, different versions of the operon, and sort of in combination of all these different mutations and wild type versions, what would be the effects? What's shown on this slide is a situation where they have mutations in the operator and mutations in the LAC-Z gene. And you get a different effect if your operator mutant is on a different DNA strand than your LAC-Z mutant. So in the left-hand side, the operator mutant is attached to a functional LAC-Z gene, and the normal operator is attached to the mutant LAC-Z gene. And what this shows is that only one of these would be allowed to transcribe. That's the one with the mutated operator. And if it transcribes, then it transcribes only this wild type LAC-Z functional, makes the functional enzyme. So instead of having this mutated code for LAC-Z, that's not the one that gets expressed. The right-hand example, though, the combination of the mutated operator with the mutated LAC-Z gene produces the non-functional beta-galactosidase, and there's no ability to transcribe this one, the functioning beta-galactosidase, because it's attached to the functioning operator that gets repressed. Okay with that? So there's a lot on here. Basically, the take-home is that the repressor or the activator, it controls only the promoter that it's attached to. So you could have two versions of genes with different promoters, and each of those promoters could act independently. Another example here, so functional and non-functional repressor molecules. So this it's kind of a combination, right? We have one that should be off in the absence of lactose, and one that should be on in the absence of lactose, right? So who wins? Which enzyme wins out? I guess one thing, I don't know if you're thinking about it the same way I am. The repressors, when they get made a lot. There's a lot of them floating around. So it's not like one single repressor enzyme gets made. The cell is making like enough of those to have around. So it's just that they exist. There's enough of them. So what's shown here is this one. It's showing one repressor, but it's active in 2 locations. That would just indicate that there's lots of repressor around. So if you have functional repressor and non-functional repressor, in a situation where the gene is normally repressed, then the non-functional ones, they just sort of don't cause any problems. They just go off and do their own thing. But the functioning repressors can get in there and repress both of these copies as long as they've got the functional operator and the promoter there. So this one would work fine off in the absence of lactose, on transcription on in the presence of lactose. So a little bit different from what we saw before. These 2 examples are, I have them labeled as partial diploids. I think that's how your book calls them. It's basically a cell that artificially has 2 versions of the Lac Operon system, you know, for science. All right, so one more. We've seen all these different kinds. We've got a mutant repressor. Now we've got an active repressor with a mutant repressor and wild-type versions of the operator. In the presence of lactose, now we've got transcription of both of these. So I think the point that they want to make here is that these could be on the same strand of DNA, the same piece of DNA, but they can act across DNA pieces. So I mentioned that we're kind of doing the simplified version of expression regulation. This is a more, gets out into a little bit more of the realistic expression regulation. There can be a lot of ways that cells respond to their environment. So it's not necessarily just so simple as I have lactose, I need to break it down. This is a situation where the Lac Operon is also under the control of other transcription regulators. And those are responding to the presence of glucose. So low glucose, high glucose. And I guess maybe starting at the bottom, so if glucose is high, then in that situation, the RNA polymerase can't attach. So let's maybe focus on the bottom one there. This cap molecule, would that be an activator or a repressor? We got one for repressor. Any other votes? All right, 2 votes for repressor. The cap molecule is a repressor. What I see is the cap molecule. Right, everybody's on for repressor. I think I'm seeing this right. So it says down here, this cap molecule is unable to attach to DNA. And if the cap molecule is not there, then RNA polymerase is also not there. So what I'm seeing is that the cap molecule is required for RNA polymerase to attach. Is that what you see? So that would make it an activator, right? So for our definitions of activators and repressors, molecule attached to DNA enables transcription, that makes it an activator. If it's not attached to DNA, it does not allow transcription, right? Okay. So this is sort of, I guess, upstream of the Lac system we've been looking at so far. So regardless of the presence or absence of lactose, the presence or absence of glucose kind of gets in there first. So low amounts of glucose, high amounts of glucose. What it says at the bottom here, basically, if glucose is high, then even without the, let's see, what do I want to say? It's kind of a flow chart will help here. Lac Operon. Glucose. Glucose. And lactose. Right, so the I'll kind of simplify it here. So off. So if there's a lot of glucose around, that basically prevents the activator from allowing transcription to happen. And regardless of how much lactose there is in the environment, this one basically stays off. So we saw previously, let's complicate us too much. So previously we saw this basically where RNA polymerase would get in here as long as nobody was in the way. And the new information we're getting about this glucose regulation is that RNA polymerase also requires an activator. And so if that's not present, it's not going to do that. So it's sort of like a flow chart. If there's high glucose, that basically prevents transcription of the Lac Operon. If there's low glucose, then we get to evaluate how much lactose there is. High lactose, low lactose. So that gives us, in this scenario, we've got 3 outcomes. 2 of those outcomes have transcription off of the Lac Operon. So either there's a lot of glucose, or there's a little bit of glucose and a little bit of lactose. We'll say maybe no lactose. So 2 ways for it to be off. So what's going on with the high glucose situation? Why would that be a good time to have transcription of the Lac Operon be off? All right, so basically, glucose is coming off as more of a preferred food source here, an energy source. So if there's a lot of glucose, then that might take priority, and it's not worth digesting lactose at that time if you're swimming in glucose, right? So either of those is going to get you energy. And I don't know, maybe it's not worthwhile to break down lactose at that point. This other situation, if there's low glucose and also low lactose, well, that doesn't make sense to digest lactose if there's no lactose around. So that's kind of a different reason to have this off. So its first reason at the bottom was you've already got enough glucose, you don't need to digest lactose. The middle reason is there's not any lactose to digest. And then this top scenario is basically where cells find themselves with lactose available, but not glucose available. And at that point, it's worthwhile to break down the lactose, right? Okay. So different ways to control expression of the same operon in this case, all for the benefit of the cell. All right, let's try this one. Do multiple choice. All right. Not too many of you out there, but maybe we can weigh in on this one. So poll everywhere. What would happen if you had these two mutations? So a super repressor and the constitutive operator mutant. Ooh, big split. All right. I like when I show you the answers and people who aren't sure about their answers change their mind. We'll go back to here. Okay, so the super repressor, if you remember, that was the form of this. That was all spiky and didn't have the ability to interact with lactose. So from the super repressor's perspective, that mutation would always prevent transcription. But we saw then also the Lac OC mutated the operator there to prevent the repressor from attaching. So it's kind of a question of which takes precedent here. So the repressor would love to clamp down on the DNA and stay there forever, but the operator doesn't have a DNA sequence that it can recognize. So it's really the operator that is the more important mutation here. It's going to prevent any kind of repressor from attaching there. So that's an always on situation. Change our answer on that one. Does that make sense? Okay, so we saw some hypotheticals about different ways that operons and genes can be regulated. This is another one that's also kind of a model for expression regulation. Um, the one I put on the assignment is similar, but we've seen examples of all kinds of different variations of these. So I'll go over this one. This will be different from what's on the assignment, and hopefully you can, as you work through the assignment, figure out yet another way these can be, genes can be regulated. So this is called the Trip Operon, and the Trip Operon has to do with building up tryptophan, which is an amino acid. And the Trip Operon, again, as an operon, has multiple genes with related functions. So these genes, Trip E, Trip D, Trip C, Trip B, Trip A, each of those encodes an enzyme, and those enzymes are responsible for several steps converting a precursor molecule, charismate, into the tryptophan molecule. So kind of handy. If the cell needs tryptophan, it turns on the transcription and builds these enzymes, and it gets tryptophan. And this is a different kind of regulation than we saw with the Lac Operon. So lactose, when we think about lactose, we're thinking about breaking down lactose as an energy source. And when we think about tryptophan, we're building up something that needs to be used by the cell. And previously, I was trying to set those as contrasting situations. So we saw lactose, Lac Operon was an inducible system. So lactose could be broken down for energy. In the Trip Operon, the charismate molecule gets built up or modified into tryptophan, tryptophan, and tryptophan would be required for protein. So I think we talked about this before. So building up a raw material as part of what you're doing biochemically in the cell metabolism. This needs to be made as long as it's being used. So maybe we'll go through a similar flowchart to what I did before. If you had a situation of low tryptophan, make more tryptophan. So turn the transcription on if tryptophan is in low supply. And then if you make so much that it's no longer being used, that's a good situation to turn off transcription. Got a request for glucose on and off. Do now briefly. One of the things I think sometimes students get hung up on is they want to make these signaling systems more complex than they are. The nice, elegant part about this is it's all controlled by tryptophan. So nothing in this pathway even recognizes the presence of chorisme, that starting point molecule. It's really just if there's tryptophan, we don't need to make more. If there's not enough, then we need to make some. And it's really the making of tryptophan that makes the tryptophan abundant, obviously. And so if you want to not have as much abundance of tryptophan, then you stop that process. Kind of another aspect of this is that this tryptophan can get used so if you are like actively making tryptophan as a cell and it's always being used, let's say you find yourself in a high energy, high nutrient environment as a bacterial cell and you're constantly making new cells and new proteins, then it might be in your interest to keep making tryptophan and other amino acids. And there's no buildup of amino acids because you're always putting them to use. And if that was the case, then you'd be kind of in this circle over here of making it, using it, having low amounts, and that signals to make more, and so on. So it's really when you are at a point of making more than you're using that this builds up and then serves as a feedback to the pathway, right? Okay, so we have transcription is enabled in the absence of tryptophan or when tryptophan is low. And then when tryptophan is present, that's the molecule that interacts with the red molecule to stop transcription. So red molecule, color-coded red, that makes it a repressor. In the absence of tryptophan, it's an inactive repressor. In the presence of tryptophan, it becomes an active repressor. So similar to what we did talking here, little box around that. So this was the repressor with the Lac Operon. And the repressor for the Trip Operon is the same principle, but in a different way, where there is the inactive repressor now in the presence of tryptophan. Oh, shoot. Sorry, I knew I was going to get something wrong here. As I think and draw at the same time. That one on the left is an active repressor in the presence of tryptophan. And an inactive repressor in the, all right, let me just make sure I said that right. Active repressor in the presence of tryptophan, inactive repressor in the absence of tryptophan. Compare that with our diagram. Great. So another version of that allosteric regulation, the tryptophan interacting with the repressor still changes its shape, except in this case, without tryptophan, it's not repressing, and then with tryptophan, then it can repress. So a nice illustration of how a repressor, depending on the repressor, can be involved in an inducible transcription system or a repressible transcription system. Which kind of brings us full circle to this idea. It's a little bit, it's a lot of, it's sort of like when you're, I guess, designing an electrical switch or something. There are a lot of ways to be on and off. And so it's helpful, I think, maybe as you're guiding your thoughts about this, to think about why the gene is on or off. So molecules that are substrates or products in the reaction that's being done. Substrates are the signals for inducible systems, so something that needs to be broken down. Products are the signal molecule in a repressible system, something that would need to be turned off if it's in abundance. So inducible, repressible, affected by substrate or product for the signal molecule, regulated by an activator or repressor. Just these questions ask you about the definition of activator and repressor. What kind of signal molecule, what happens when the signal molecule interacts. So maybe one way to think about this, you could draw a big old table, all these yes or no possibilities. So I guess you could connect the dots here and say we've seen the Lac Operon system was controlled. Let's see, it was inducible via a repressor interacting with the substrate, right? And then the Trip Operon was repressible, interacting via a repressor, interacting with the product of the reaction. Connect all these together. And as we've seen, just the simplified version that we do here, repressible really goes along with systems that are interacting with the product. And then that other question is transcription on or off, depending on whether the activator or repressor is attached to the DNA. That's another thing you should be familiar with. So lots of 50-50 questions. But I think if you kind of understand how it all connects and why, really. So why does the cell need to turn something on for transcription or turn it off for transcription? I think that helps you to maybe put all the pieces into place. Questions on that? All right. It's just so quiet out there. I think I just want to stop at this point. So we got the assignment 7. I just put the due date for Thursday of next week. If you want to do that, give it a shot. And if you have any questions, we can go over them on Tuesday. We'll do a little more review of this kind of stuff. And then next week will be our last 2 classes. We've got the Tuesday topic will be gene expression in eukaryotes. And then Thursday, we'll wrap up if we have anything left to cover. And then there'll be some time to review. And it'll probably be toward the end of next week when I have the practice questions up. But there'll be a week between our last lecture meeting and then the final exam is the Thursday of the final week. So of Thursday of finals week. So they should have still some good amount of time to get practice questions and work through those for the final exam. Any questions on grading, scheduling, topics, anything? Okay. Well, I guess we can be done early today. Have a good weekend. I'll see you on Tuesday.